Very good morning to one and all present here. We welcome you on behalf of Sri Ramakrishna Institute of Computer Science and Education and Apparent Pharmacy Medicine to the day one of the national webinar on the COVID-19 issues and challenges. Now I request to Dr. Jay Bargi to continue the session.
Good morning, all. It's my pleasure to welcome you all in the webinar on COVID-19 issues and challenges, organized by Sri Ramakrishna Institute of Computer Education and Applied Sciences, henceforth we call it SRKI, in collaboration with Operant Pharmacy Federation, henceforth we call OPA. Myself, Dr. Jay Vargi, in your today's host. I'm heading the Biotechnology Department of SRKI. I've done my MSc and PhD in microbiology, and I'm a teacher in the SRKI since last 17 years. This webinar is intended to give the brief overview regarding the present status of COVID-19 pandemic to all the undergraduate students, postgraduate students, research fellows, faculties of all life science subjects as well as pharmacy subjects. We will be discussing all aspects, possible aspects of COVID-19 pandemic, like the pathophysiology of the disease, the diagnostic part of the disease, management part in terms of vaccine development and commercialization, and how the environmental response is influencing the spread of the pandemic. So this is what the scope of the webinar is. Sri Ramakrishna Institute for Computer Education and Applied Sciences is a leading science college in the South Gujarat region. We are affiliated to UGC recognized Veer Narmad South Gujarat University. We offer BSc courses and postgraduate MSc courses in biotechnology, computer science, environment science, and microbiology. We also offer postgraduate diploma in medical laboratory technology, and we are proud to give such co-curricular and extracurricular exposure to the students of our institute as well as to the students of South Gujarat in general. With me today is Mr. Vikram Chaudhary. Mr. Vikram Chaudhary is the founder and director of Operant Pharmacy Federation. We call it OPF. Now, OPF was established to serve the community of students, researchers, and faculties of pharmacy subject. OPF follows all ethical pharmaceutical standards and norms and is primarily focusing on all aspects of pharmacy profession. OPF explores the different facets of pharmaceutical profession and industry by supporting the causes and issues related to pharmaceutical education and other healthcare sciences. They cover all the major facets and aspects of pharmaceutical profession with the ever-inspiring vision of Team OPF as well as of Mr. Vikram Chaudhary. I I welcome Mr. Vikram Chaudhary. He will be guiding us through these 2 days as a moderator of overall webinar. I thank him in advance. Our today's first session will be conducted by Ms. Avni Kaushal. Ms. Avni is a dynamic and versatile research scientist with more than 10 years of extensive research experience. She possesses wide-ranging knowledge in the subjects like cell biology and protein characterization methods. She is having good analytical and problem-solving skills. Currently, Ms. Avni Kaushal is working as a scientist at the Vaccine Research Center of Kedila Pharmaceutical Limited, Ahmedabad. As a scientist there, she manages challenging research schedule in highly complex and ever-demanding research environment. Prior to joining as a scientist at Kedila Pharmaceutical, she has worked as a scientist, as a research fellow at institutes across India. She had a brief stint as a faculty in microbiology also. Ms. Avni has published a few really good quality research publications in high impact factor journals. She has completed her BSc as well as MSc in biotechnology, BSc in microbiology and MSc in biotechnology from Sardar Patel University, Vallabh Vidyanagar. I welcome Ms. Avni Kaushal for her first talk of the session, and the title is COVID-19 Vaccine Development and Commercialization. I hand over the webinar now to Ms. Avni Kaushal.
Good morning, everyone. Thank you for a wonderful introduction, sir. My name is Apni Kaushal, and today I will give a little insight about vaccines, how they are developed, and till their commercialization. So I'll start with sharing my presentation. Today, my topic is vaccine, its development, until commercialization. I'll begin with the outline on the topics that I'm going to talk today, beginning with the introduction of vaccines and its types. So, what exactly is vaccine? The word now is becoming so common due to the pandemic of COVID-19. Basically, vaccines are preparation of killed or weakened microorganisms that can be given to a person orally or injected in order to prevent a certain disease. So when Edward Jenner in 1796 started working on vaccines, he worked against smallpox, and thus the term vaccination was coined by Louis Pasteur in 1881 as in honor to Jenner. Why are these vaccines important? We have drugs, right? We already know that drugs can cure any disease that we know. But then why are vaccines important? So vaccines can prevent the spread of disease before even you have that disease. So the vaccines, they prevent highly contagious, dangerous, and deadly diseases. So rather than waiting for a drug to come after you have the disease and after you have the symptoms, a vaccine can prevent them. Um, those vaccines which are included in today's infant criteria are measles, polio, mumps, chickenpox, whooping cough, diphtheria, and HPVs. So currently, what we know is there are more than 2.5 million children under the age of 5 which die from these kind of diseases, which can be prevented by using vaccines. The vaccines not only benefit a single individual, it benefits the entire community. Like when we talk about polio, when we talk about diseases like polio and the current one, COVID-19, the entire community is hindered by such a small virus. So when we say that a drug can solve the question of a disease, a vaccine would not only help the individual who has this disease, but to the entire community who are suffering for the same problem. So today we can say that it has reduced the prevalence and incidence of many diseases, and it has also eradicated diseases like smallpox and polio. How does WHO, the World Health Organization, define vaccine? It says that a vaccine is a biological preparation that improves immunity to a particular disease. A vaccine typically contains an agent that resembles a disease-causing microorganism and is often made from weakened or killed form of microbe, its toxins, or one of its surface proteins. The agent simulates the body's immune system to recognize the agent as foreign, and it destroys it and also remembers it so that the immune system can more easily recognize and destroy any of this microorganism when it later encounters it. The milestones that we have achieved since 1796, when the smallpox vaccine by Edward Jenner was introduced, till date, you can already see on the screen that we have improved quite a lot from the year 1796 to the oral polio vaccine that you can see in 1962, which is still being given to all the infants every year. Then there is MMR vaccine. Then after 1990, the vaccines have taken a turn. It is now a mandate in our country to give vaccines to the infants. I'll go about the types of vaccines that you already know. So in your curriculum, you've already studied all the types of vaccines. The vaccines that today we include in our topics are live attenuated ones, virus like particles, recombinant bacterial vectors, DNA based recombinant viral vectors, recombinant subunit viral proteins, synthetic peptides, and whole inactivated virus. So on the right, you can see the examples of the vaccines which were like introduced recently. So live attenuated vaccines, you can see that tuberculosis, oral polio vaccines, measles, rotavirus, and yellow fever. For inactivated or killed antigen vaccines, you can see pertussis and polio vaccines. Then for subunit ones, you can see again acellular pertussis, then HIV, pneumonia, hepatitis B. For toxoids, you can see that tetanus toxoid and diphtheria toxoid are used for vaccines. Next, we will go into vaccine development now. So, how do we decide on whether we want to develop a vaccine for a certain disease? So, we cannot just say that we want to create a vaccine or develop a vaccine for every disease that we know about. So, we have to think about the scientific feasibility. What is the disease burden? What is the public perception for the same? And then we go ahead with the vaccine research. So the basic research begins with thinking. A lot of thinking we put into whether we should go ahead with the vaccine development or not. So the current status of COVID-19, as we all know, that the vaccine is needed, and the reasons for the same are that even if we have the drugs for COVID-19, the problem that occurs with it would be that CDC has already declared that the memory cells for the disease last only for 3 months. So after 3 months, the person can again suffer for the same disease. So that is why the vaccine has been suggested for the same. So how does the vaccine development work? So the vaccine development is a very long and a complex process. It usually lasts for 10 to 15 years. And it is the involvement of public and private companies is combined, and then the decision for the vaccine development begins. So the current system of developing and testing and also regulating the vaccines was developed during the 20th century as the groups involved standardized their procedures and their regularization. So vaccine development and testing follows a standard set of steps. So anything that we generate, whether it is a vaccine or a drug, it has to go through certain steps. The steps are being regulated by agencies either of different countries or WHO itself. So the first stages of any vaccine are exploratory in nature. That would include R&D and other few steps, including the preclinical steps. So the regularization and oversight increases as the candidate vaccine makes through its way through the process. So the next slide are the different stages of vaccine development and its testing. The phases of vaccine developments begins with exploration, then the preclinical step, then the clinical development, regulatory review and approval, manufacturing, and quality control. So I'll be explaining all the steps from the next slide. So the exploratory stage. So this is a basic laboratory research which often lasts for 2 to 4 years. So it is normally funded by academic or government scientists. It identifies natural synthetic antigens that might help prevent or treat a disease. These can be many different candidates. It doesn't have to be single candidate. These antigens could include virus-like particles, weakened viruses or bacteria, weakened toxins, or other substances derived from pathogens. So basically at this steps, the candidates are many, and then we segregate or we reduce them slowly by going to the other stages. The next stage is preclinical stage. During this stage, the studies are being conducted on cell culture systems or animal testings. So the candidate vaccines is being tested for its immunogenicity or how it can provoke an immune response in that being. So basically, the preclinical evolution, it is a compliance of good laboratory practices, then production characterization and quality assurance of a candidate vaccine, toxicity and safety testings, potency, immunogenicity, how the adjuvants and additives that we add to it works. So I'll go to the next stage. Um, Now, after the preclinical stage, there are many phases to a clinical trial. Phase 1, phase 2, phase 3. Currently, due to COVID-19, you might have heard about all these three stages. It's in the news, it's in everywhere. So I'll just give you a brief what exactly these phases are. So basically, phase 1 denotes to safety and immunogenicity genicity studies, which consists of adults of about 20 to 80 adults for the study. So the human participants at this stage are less. They are only 20 to 80 or most probably 100 of them. The next stage, the phase 2, it consists of almost 100 to 300 participants, and these are for safety, dose ranging, and studies. The third stage, which is a large scale study for participants from 1,000 to 3,000. So this is how the studies for the three trials goes on. So FDA, after reviewing all three stages, would go for the approval steps, which also takes years. The phase 4 is not always present in the study. Phase 4 consists of additional parts that can be studied if the three stages don't include those steps. Like if we want to study for infants, so when we study for the phase 1, phase 2, phase 3, those studies are being carried on adults. So phase 4 can include the studies for infants, for different age groups, for different kind of um, demographic and ethnicity of those people. So the next slide, I'll tell you in detail what phase 1 vaccine trials include. So phase 1 vaccine trials includes humans of small groups, basically adults, and the vaccines, if intended for children, will be tested later on. The phase 1 trials are usually non-blinded. That would mean the person who's getting this vaccine would be knowing that they are given this vaccine or whether they are given the placebo instead of it. The goals of phase 1 testing are to assess the safety of candidate vaccine, to determine the type, extent of the immune response, and it is usually a small minority group. So it may or may not use a challenge model. The challenge model is usually after giving the vaccines, they are given the version of pathogen itself for the challenge to see whether the vaccine is working or not. After a successful phase 1 trial, we will proceed to the next stage, that is the phase 2 trial. It is of several hundred individuals, and some of the individuals may belong to the groups containing this disease or having the risk of this disease. These trials are randomized and well controlled, and they also include a placebo group. The goals of phase 2 testings are to study the candidate's vaccine safety, immunogenicity, proposed doses, schedule of immunization, and method of delivery, which are all very important at this part. After a successful second trial, they go ahead with the stage 3, phase 3 clinical trials. So with the stage 3, the trials go to the extent of a very large scale. They go into the thousands of people are recruited for this, and these are randomized, double-blind, and involve an experimental vaccine being tested against the placebo. Now, what the placebo can be a saline solution or a vaccine for other diseases or some other substances also. The basic goal for phase 3 is to assess a vaccine's safety in large group. And if there are any side effects, the rare side effects, which cannot be seen in smaller groups, they can be tested here. Later, after phase 3, there are regulatory approvals and licensures which have to be taken into consideration before applying to agencies like FDA, DCGI, or EMA. So after the phase 3 trials, the vaccine developers now applies to the agency of different countries. For countries like US, the licensures are to be applied to FDA, which has a very stringent method of approving these vaccines. They will review the manufacturer's test lots, then they'll check for the potency, for the safety, for the purity, and they might also conduct their own testings of the manufacturer's vaccines. For India, the regulatory agency is DCGI. For European countries, it is EMA. Now, after the licensure, what happens is that there are many different trials which need further studies, for which we can go into phase 4 trials. The phase 4 trials usually is for testing the safety, efficacy, and other potential uses of the same vaccines. Now, the timeline. So we've already talked about how the discovery, exploratory, preclinical, and different clinical stages or phases works. So what exactly is the timeline for a normal vaccine to cross all these phases? Usually from the discovery to the manufacturing preclinical stages, it takes around 3 to 8 years. For phase 1, phase 2, phase 3, it usually takes 2 to 10 years. And for the regulatory reviews, it also takes 1 to 2 years. Going ahead with the manufacturing, so after the approval, there are still steps which takes around 24 months, around 2 years to complete. So that includes how we basically go into a manufacturing after getting all the approvals. So it begins with raw material reception. The incoming of all the raw materials are checked, and then the quality specifications are maintained for the same. Then we go for bulk antigen manufacturing, a formulation part, filling part, packaging part, and lot and release part. After that, it has to be shipped and distributed. So usually a quality control is set up at every kind of step, and these sites can be different sites, as in the raw material reception and the bulk antigen manufacturing can be at 2 different production sites. The formulation, the filling can be at 2 different production sites. It doesn't have to be in the same company. So, for example, currently you might have heard that Serum Institute is going to produce the vaccine which has been in phase 3 trial from Oxford University, the one which has been collaborated with AstraZeneca. The production can be done at Serum Institute, which is situated in Pune, India. So even though the entire trial was taken in different countries, the manufacturing units can be in different countries. Next, what are the challenges that a vaccine development has to overcome? So the manufacturers are challenged by the process, and the stringent quality control takes several months to years from the time the production is initiated. As in, if we go into the manufacturing after all the phases also, then it takes up to 3 years for the production to initiate anything. So vaccine production or manufacturing, it has a very high capital incentive. For making a new facility, it takes around 5 to 10 years. And then the investment goes from $10 to $100 million. And very highly skilled and trained personnel are essential for these kind of vaccines to manufacture. The compliance have to be from the local regulation and international regulations too. And the post-marketing approvals can take up to 2 to 4 years. Next is that the classical versus COVID-19 vaccine in the timeline format. So we've already talked that the preclinical phase 1, phase 2, phase 3 takes up to 10 years of different trials. But for COVID-19, since it is an pandemic, since it is an emergency vaccine created, so the timeline has been reduced. As you can see, the clinical trial only took around a month. The phase 1, including like dozens of volunteers, was given only 6 months. The phase 2, 6 months. The phase 2 and phase 3 for the same were combined in many of the tests, and the approval manufacture uh, will be taking more 6 months. So you can understand that it will take around 1.5 years to form an emergency vaccine too. So COVID-19, the vaccines were the beginning exploratory stage begin around January 2020. So you can expect the vaccines around December or next year, like in 2021, the first quadrant, hopefully. Next, I would like to share the information how many vaccines for corona or COVID-19 are currently in which phases. So there are about 19 vaccines in phase 1, 13 vaccines in phase 2, 4 vaccines currently in phase 3, and 1 at the approval stage. So these are different phases in which currently all the different universities, companies are trying to uh, get their clearance for their vaccines. So you can see currently the vaccines in phase 1 are CanSino. The vaccines from Oxford universities now have crossed stage 4, and there is also different institutes which are trying for the same. Serum Institute phase 2b is also trying for its own vaccines other than the production that it is taking from Oxford University vaccine. So this is all I would like to talk. Thank you very much.
Well, first of all, I would like to thank Ms. Avni Kausal for her talk. To summarize, Ms. Avni started with the introduction of what vaccines are, what is the history of vaccine. Then she explained that what is the standard method of developing a vaccine, what are the standardized steps of developing a vaccine, stages, all the stages she explained in detail. Then a bit of challenges, particularly regarding the COVID-19 vaccine. Then she explained, I liked that point very clearly and liked it much, that how for the COVID-19 candidate vaccine, the entire timeline, which takes as long as 10 years, was shrink. So how for considering an emergency situation, how the timeline was shrink. And lastly, she explained regarding the current status of all COVID-19 vaccine candidates. More than 19 vaccines are in the stage 1 and what are the advanced stage vaccines. I hope that the candidates which are in phase 3 as well as at the approval level, 5 of them, may get approval as fast as possible. This lecture will give us idea regarding that we are now very close of this controlling the pandemic. It's a hope to each and everyone who is really fed up with the entire scenario. So at the end, again, I would like to thank Ms. Avni Kausal for a wonderful lecture.
Thank you, sir.
Now, there is a live chat of YouTube. So if there is any question in the live chat, I think Vikram sir will pass it on to the Avni ma'am and we can answer. So I don't think there is. there's any question at present in the comment section.
Okay.
On behalf of all participants who have benefited from this session, I would like to thank again Ms. Avni Kausal. Now, we'll have a second talk in the today's session, but before we can go to the second talk, we'll have just a 2 minutes break in terms of changing over, as well as uh, yeah, there is a question before I can leave. There is a question, and the question says, even after all stages, is it any chance that vaccine may get some problem, or there is a change in the basic nature of virus, so as vaccine is rendered ineffective? Is it any possibility like this?
No, there
Or
are many stringent steps taken for the same to see to it that the candidate which reaches the final stage doesn't show any problem. So that is why there are regulation at every step for the same. Sometimes it happens that some demographic or some ethnicity has an issue with the same. Like with the rotavirus candidate, it was being developed in US, and the same vaccine was not working on the infants in South Africa. The problem occurred due to demographic reasons. So, when we considered a clinical trial at larger stage, we considered all the demographic and ethnicity groups. If it has been trialed on everyone, then and only then we can say that the vaccine would work for the same. So, when reaching the last stage, it usually crosses all these steps, and then usually the problems like this do not occur. But it's a very good question. Thank you for asking.
and there are a lot other questions coming on, ma'am. So kindly hang on. The second question is that Ministry of Human Resource Development, now we call it Ministry of Education of Government of India, as well as Science and Technology (ICMR), they are organizing what is popularly now called as hackathons. So, is there any fruitful contribution came out of hackathon in the vaccine development field?
Yeah, I'm sorry, I do not really understand this question.
Now, there are specific competition events organized in which students and research fellows participate. These are organized by Ministry of Science and Technology in India or the Ministry of Education. Is there any contribution coming out of such instant competition in terms of vaccine development?
I have no idea. I'm so sorry.
So, if you ask me, as per of my knowledge, all the vaccine candidates which are in preparation in development phase in India, none of them have came out of hackathon. These are from the research of the respective pharmaceutical industry or the
Yes.
vaccine organization. Now, we'll have a second question, another question. Uh, is there any vaccine prepared by India for COVID-19 currently, and is it working on?
Yeah, there are actually two basic companies which are forming vaccines in India also. One is Bharat Biotech, the other one is Serum. The third one is Zydus also.
Yeah.
Zydus. All three of them have already good candidates, vaccine candidates, which are into phase one, phase two, or phase three trials right now. Phase two, I guess. Phase three, they have not reached yet. But there are other companies outside India which have crossed phase three, are into the final approval stage, and for which the production will also be conducted in our country. So overall, our country is in good hands, it's in good shape, and we might get a vaccine very soon.
Thank you, madam, for the answer. There is another question that is there any specific change being made in the regulatory procedures by the government of India or any other government, specifically keeping in sight the COVID-19 vaccine? I know that you have covered this part in presentation, but the question has
Yes.
been asked: that is there any regulatory changes being done?
Yes, due to the emergency vaccine regulation for COVID-19, yes, they have made many changes. Yes, the COVID-19 vaccines would be considered as priority. So the approval, the basic steps would be reduced. The phase two, phase three trials might be combined and taken, the trials would be taken together. they have cut short with the approval methods, with the speed that they take everything. Like the timeline takes 10 years for a normal vaccine, but for the emergency vaccines, it cut shorts to 1.5 year. So they have taken many different steps. Plus, there are news that ICMR and the government is opening a new virus vaccine, some new website for it, which would be open like in few months. It's a portal. It's a new portal that they have defined only for vaccines in India for the first time.
Now, ma'am, many people have asked directly to me. There is a question in the chat also that virus is something which keeps evolving, keeps changing, and there is a lot talk regarding mutations in the virus. And
Yes.
then there are reports that in different countries there are different strains of virus. So the virus with the high mortality in Europe had a different genetic makeup, and the viruses in China and India are different. So how a single vaccine candidate will address all this broad range of mutated viruses? That's one of the questions.
So I would say that it is true. The mutation does occur. So you might have heard swine flu, which was a recent danger few years back when everyone was talking about H1N1 flu, and that the flu vaccines have to be generated every year. So you can imagine the pressure on the vaccine production and the manufacturing unit. Every year, the strain changes. So WHO recommends some strains for northern hemisphere and southern hemisphere 6 months before we can start the current year. So they give us the strains, they give us the names, and we have to start the production and the research and every trial thing, everything within the 6 months, and then produce the vaccine for the next year. So what happens is, what becomes the difficult part is, yes, the mutagen, it is called antigenic drift. So everyone from the biology background already knows that the antigen on the surface of the virion is what actually causes some sort of reaction or the disease that we currently know as. The symptoms are due to it. So the antigenic drift is that it is a gradual change in the, it is a shift, a genetic change in the viral itself. So, due to that reason, we see many mutation. The mutation can occur from one human to another human for demographic change, for any kind of change. So, the consideration that we took for flu a few years back, that we need to have a vaccine every year, we are considering the same thing might happen with corona if the mutation occurs very severely. But since CDC and WHO has already announced that the vaccine which comes once will work for the entire time, we are hoping that this kind of situation doesn't occur for coronavirus. We still
Okay,
don't know what will happen.
yes, thank you, ma'am. And the next question is, if a successful vaccine candidate is developed and manufactured, then what is the role of WHO in the process of dissemination, spreading of the vaccine?
The WHO has to approve that candidature of the vaccine, and then WHO would recommend to different countries whether this vaccine candidate would be applicable to them or not. As I told you, that the ethnicity and demographic reasons are very important in a vaccine. It might work for Asians, it might not work for Caucasians. So WHO would approve on the basis of what FDA, that is US based agency, that BCGI, that is India based regulatory, and for the EU it is EMA. So accordingly, it will recommend in which country which kind of vaccines are approved. So even if you pass all the stages, even if you have manufactured a vaccine, then WHO has the last hand. Like, it would decide whether it will work for your country, whether you can take it or not.
Now, if I understood the second part of what the wanted is that how the vaccine will be distributed to different countries and how it will be provided. There are some organizations like Gavi, which is associated with the distribution of vaccines to different countries. So there can be a role of other organizations besides WHO in this. Thank you for the answer again, ma'am. The next question is that what will be the influence, side effects, if any, of taking the vaccine? For example, if we are giving the polio vaccine to some kids, they say that mild fever will be there. So can there be some side effects or something like this of taking the vaccine?
So even if there are side effects, we might not come to know right now. It might take us 10 years to know whether this vaccine has major side effects or any kind of problem, which might occur later, like from generation to generation, whether it passes, whether the side effects are really harmful. But currently, what we are working on is just safety issues at on the spot, taking the vaccine doesn't cause us rashes or any other severe kind of symptoms. That is all we can do for an emergency vaccine right now. That is why usual vaccines, the original classical method, takes 10 years to develop a vaccine to test all the side effects and everything. But for emergency, we cannot say what kind of side effects might be there.
And then there is a question again repeated by more than one viewer. It is regarding that what will be the length of memory or the immunity provided by the vaccine in terms of, do we need to have booster doses? Do we need to take the vaccine in more than one doses?
So what we have heard is that yes, you might need to take booster doses. It might be a two-dose vaccine, but currently none of the companies are declaring themselves that how it would work. It is only due to the clinical trials ongoing that we have heard that it might take a booster dose. But hopefully it won't be every year. It might be once, and the second dose might be within the 3 months, and that would be it. And we are hoping for the same thing, like flu, not every year, hopefully.
Okay, and then one question is what are your observations regarding development of herd immunity without vaccine in India? What is the
So the
status of
herd immunity works on diseases or viruses like chickenpox, so which do not cause death in severity. So herd immunity for corona would not work because uh, the people having high diabetes or blood pressure or are very aged tend to have severity of this disease. So, according to my observation, herd immunity won't work for corona. But maybe when the vaccine comes out, maybe if half of the community is vaccinated, the other half might not need it. But I'm not very sure about it. The herd immunity doesn't work in this disease.
Then there is a question that there are news coming in, pouring in the media regarding the new strains and species of viruses, specifically from the China. Yes, the sources are unconfirmed. These are only media speculations. But what are your views regarding new strains of the same virus coming in?
So actually, yes, I've seen them in the news, but we cannot say anything till any official body declares whether these mutations have occurred or not. Yes, there are different strains, like the one which were present in Italy were different from which were present in US. That is why they say that the death probability was more in some countries and not much in others. Even in India, we've seen that the many different cities have different kind of death prevalences. So in Delhi, in Ahmedabad itself, the deaths were quite a lot. So the strains, they said that was a very big difference in both. So, but still, none of the major authorities or government bodies are declaring the official statement on this thing. So we cannot comment on it. I'm so sorry.
And again, there are two questions from the different individuals, and there is a lot speculation in the public in general also that because we have released the regulations as well as since the timeline of vaccine development, are there any flip sides of this? Are the vaccine quality and safety been compromised? Is there any risk of creating vaccine in haste?
So there are always risks with such things. Although they have cut down the timeline, the regulatory bodies are still stringent with what is formed. The only problem that I can foresee is that the side effects which come later on, like the one which
Long
are not
long run,
visible,
yes.
which are very rare and can be in a very small group of people, which we might not come to know right now. So, other than that, it is still stringent. It is only in a fast scale.
I feel that suffices the question answer concern.
Thank
And
you.
I again thank Avni ma'am for satisfactorily answering plenty of questions of the audience. Thank you again, ma'am. Uh,
Thank
we'll have a second session, and a feedback link will be shared at the end of the second session. And all the participants need to fill the feedback. Those participants who have registered for the event and have filled feedback for both the days, for today as well as for tomorrow, they only will be eligible for getting the certificates. So all the questions regarding certificate will be sent to your email ID only after we receive the feedback of both days as well as registration. We'll manually check it and then we'll see the certificates for the same. This is just to ensure the active participation and so as the webinar is worthy. The purpose of webinar is not to spread the certificates about spreading the awareness. So I sincerely request all the audience to just fill the feedback form. It will be shared at the end of second talk today, not now. So you need to fill in the feedback form because there is a common feedback form for both the speakers. Secondly, Operant Pharmaceutical Federation, as I told in my introduction, is actively engaged with the developments in the education research of pharmacy and other healthcare sciences. Is organizing a week-long event known as Pharmanesia 2.0, and this year it is E event, so it is Pharmanesia 2.0 E. This event will include such webinar sessions, informative sessions from the speakers all around the world. Plus, there will be competitions for the health science and pharmaceutical science students, competitions with plenty of prizes to win. So, I would request all the participants to register for the same. The link and the details are there in the comment section. So, again, a request from our side to participate in this week-long event. I thank Avni Madam for being with us. We'll have a 2 minute break between two talks and then we'll resume ourselves with this webinar. The second talk that's regarding the diagnosis of COVID 19, the present status and the future of the diagnosis.
Thank you, sir. I want to add on the drug discovery hackathon. One questions arise from one candidate. Uh, Drug discovery hackathon, which organized by MHRD Innovation Cell. This is especially for the COVID-19 specific drugs and some projects going on there in CSIR, CDEC or NIPER, or they are having some problem statements. So they want to solution of these problem statements. So they have opened the problem statement for each and everyone. The official registrations are open for the drug discovery hackathon. Those candidates who can participate in the drug discovery hackathon, who having any idea about the drug discovery or vaccine development, they are more focused on the computer aided drug design. Okay? They are focused on the molecular docking, molecular modeling, pharmacophore mapping, and QSR studies. So, interested candidates, they can visit the website of innovateindia.mygov/DDS2020. So they can participate in the drug discovery hackathon. This is officially and only for the COVID-19 problem statements. For phase one, registration is open till 30th of September. And for Pharmanesia 2.e, Dr. Jay Varghese is already informed to you. You can visit the Pharmanesia 2.e for the more information. Thank you. Over to you, sir.
We'll have a 2 minutes break, sir, and you can have that video for that.
Sir, shall you take the 2 minute break or in the same speed
We
we
are
will be
having
continue the session?
now we are we'll continue session, but after 2 minutes break, you can play that video regarding the break, the information.
Yeah, yeah, sure, sir. I am playing the AV.
Yes, thank you. Thank you, Shankar. So we are going to our second talk of the week, uh, welcome again, all of you. And in this talk, I will become a we discuss about the diagnostic part of the disease, we talk about the management, the part of the disease. presentation. So we are discussing COVID-19 diagnosis, current and future strategies. Let me start with the details regarding the differences in terms of tests carried out for the COVID-19. Now, all the tests which is carried out in the diagnostic laboratories fall into the three major categories. The first group of tests will target and detect the virus RNA, and these are the gold standard diagnostic tests for the COVID-19 is prescribed by regulatory agencies like in India ICMR. So if you want to diagnose the coronavirus presence in a person, if you want to say that person is corona positive, then the definitive gold standard method is always detecting the virus RNA. This is the first group of tests. The most common test in this group is what is popularly known as RT-PCR. The second group of tests is regarding the rapid antigen detection. Now, when we say antigen detection, then technically it is the detection of virus protein. So it is detection of virus protein that is going to be our second group of test. The third group of test is going to be the detection of antibodies against the coronavirus in the serum of the patient or individual. Now, this is not for the diagnosis technically because the antibodies are even there once the person recovers from the disease, what we call the convalescence stage. So, in the convalescence stage, the antibodies are still seen. This is usually done to see the sensitivity of the person against the virus. If a person is already having serum antibodies, then he is resistant to the virus. This explains us the level of immunity in the society regarding against the coronavirus. So it's about what is the status of disease in a particular community. Now, the rapid antigen test as well as the serum antibody test, both of these tests are simple. They don't require sophisticated instrumentation and can be carried out close to the patient near the caregiving facility like hospitals or clinics. So we call them point of care test POCTs, both of these, rapid antigen and serum antibody. But the first test, that is viral RNA detection, is usually a complex and sophisticated test, requires advanced instrumentation. Usually the sample is collected and sent to the specific laboratories for this. Now, samples for the coronavirus detection, mostly it is either the sample known as nasopharyngeal sample or nasopharynx specimen collection shown in the left of the image. So a swab, a long cotton swab is used, and from the back of the nasopharynx, the sample is collected. The second one is known as oropharynx swab. Here, by depressing the tongue with using again a long swab, the sample is taken from the sides of the oropharynx and from the terrace of the oropharynx. So these two are the common samples. However, some home use kits in US, they are using short swabs, and samples are for such kits are taken from the outer nostrils of the person. This is how sample is collected presently in India, and most samples are oropharynx samples. However, there is a method in development. Now, this is a futuristic thing. A kit is already in development and it has already got emergency use authorization, EUA, what we call, in Europe, which uses direct saliva for the test. So, virus present in the saliva is collected in a container by spitting, and from the spitted saliva, the virus RNA is isolated, extracted, DNA is created from the RNA, and the test is being performed. This is already authorized. So the samples can be either the oropharynx sample, nasopharynx sample, sample from the nasal cavity, and the fourth one is saliva. For the rapid antigen test as well as for the rapid antibody test, the blood is the sample which is collected by the normal routine method. Now, the gold standard of the diagnosis presently today, as identified by the WHO, in India ICMR, is a technique called QRT-PCR. I'm aware that most of the students are from the biology background and pharmacy background, so they are aware of what RT-PCR is, but this is a very rapid primer of what RT-PCR is. So in order to understand how virus is diagnosed, let's first understand the structure of virus. So virus has got a helical, here you can see the red and yellow color, helical, what we call nucleocapsid. It is nothing but RNA, which is of yellow color in the image, bound with the capsid proteins which are red color and attaining a helical shape. So there is a coil like what we call nucleocapsid involving RNA as well as the capsid proteins which is enclosed in the blue color envelope. And then there are spike proteins of purple color on the surface of this blue color envelope. Now, for the definite diagnosis of virus, we don't look into the envelope, we go deeper into the nucleocapsid, and the yellow color RNA is being detected as the target site for the QRT-PCR. The red color capsid protein, specifically a protein known as N protein, is the target for the rapid antigen test. So if it is a rapid antigen test, we detect the N protein. If it is RT-PCR, then we detect the RNA. Now, what is PCR? So PCR is a laboratory technique which amplifies the availability of the DNA in the sample. So whatever DNA is there in sample, only a specific region of that DNA is increased in terms of its concentration. This is what we call amplification. Now it is involving three steps. In the first step, there is a denaturation of the double-stranded DNA. So two strands are separated by increasing the temperature. Once the two strands are separated, the reagents added into the reaction involves a primer, here shown in the green color, a very short stretch of DNA that will bind with the two separated copies of the sample DNA. In the third step, the temperature is again increased in the second step in order to bind primers with the DNA, green color part with the blue color part. Temperature is reduced. In the third step, temperature is again increased up to 72 degrees centigrade, which is the optimum temperature for the enzyme. The enzyme is known as Taq polymerase usually, which carries out DNA replication, and instead of one copy of DNA, now you are having two copies of DNA. Now this is known as first cycle, and these cycles are repeated again and again, and you will be having a specific region of DNA increased in concentration as high as 10 to 9 copies after the end of 30 cycles. So this is what we call PCR. PCR is nothing but specific part of DNA is increased in terms of its concentration. Here the key is that specific part only. Now if those sequences are absent, then the DNA amplification will not be present. So whether your DNA gets increased and detected depends upon the sequence, and these are virus specific sequence. So if my sample will have virus, the DNA will be amplified, will be seen. If my sample is negative, there is no virus, then this entire reaction will not take place and you will not get any result. Now, routine polymerase chain reaction (PCR) is an end process detection method where you detect the product at the end of process. But that can be even turned real time, and you can check the product even when it is made. One thing. Second, the routine PCR starts with the DNA, but as seen in the virus structure, but virus is not having DNA at all. It is having RNA. It's a virus which is having RNA. So what we will do, we'll first make DNA out of the RNA. Now that is known as RT-PCR. Here the RNA is available from the virus shown in the blue color. It is attached with a primer, second step shown in the green color. And then there is an enzyme called reverse transcriptase, which will synthesize DNA out of the RNA. The process is known as reverse transcription because in nature, when we are making RNA out of DNA, the process is transcription. This is the reverse process. You are making DNA out of RNA, so the process is reverse transcription. The enzyme is therefore known as reverse transcriptase, and the process is in vitro. We are doing this thing in test tube. Now, first the one strand of DNA is made, shown in green color, and then the second strand of DNA is made. So you are having two double-stranded, you are having double-stranded DNA, which can be then subjected to the regular PCR shown in the previous slide. So this is known as reverse transcriptase PCR or RT-PCR. Now, as I said earlier, you can detect the concentration of product made by what we call the qPCR. Q is quantitative PCR. Now, if I want to see how much of product is made from the sample, what is the amplification level, then besides the two primers shown here in red color, you need to have a reporter probe which is shown in the blue color. So what happens? Your sample DNA shown here in black color, as the process of PCR will take place, the probe will be releasing a dye shown here as a R, green color R. So more your DNA is replicated by the PCR, the more dye will be released, and that can be then detected by a machine. So this is what we call the quantitative PCR. It will give you the idea about how much of product is made. Okay, and the product concentration can be seen in the graph. Now, for COVID detection, the RT-PCR workflow involves many steps. It always starts with the first swab. It starts with a swab taken from the patient. Then the swab is dipped, immersed into what is known as viral virus transport medium. It will be, अगर आप इसके research article पढ़ेंगे, then in research articles it is acronymed as VTM. So VTM, virus transport medium, में the swab is submerged. From VTM, the virus is extracted, sorry, the viral RNA is extracted from the viruses. So you are having virus and from it the RNA is extracted. This is the first step, RNA extraction. Then using the enzyme reverse transcriptase, RNA is converted to the DNA, second stage. In the third step, there is a quantitative amplification of DNA made out of RNA, which can be seen in the fourth step as an output of graph. Now, if your sample is positive, then you will have the signal beyond a threshold level. Now, if your sample is negative, then the signal will be there, of course, but it will be below the threshold level. This is known as QRT-PCR. Now, this is considered as the gold standard. In order to perform QRT-PCR for the COVID diagnosis, you need first step RNA extraction. There is a ready-to-use kit available for the day, and then you perform the remaining part of PCR in a 96 well microplate, and amplification, the temperature up and down required for that, will be carried out by a machine known as RT-PCR machine or qPCR machine. Now, in this qPCR machine, the temperature will be changed for the step 1, step 2, and step 3. And the same machine will also detect the signal which is released with the DNA amplification. An example of qPCR machine is shown here. It is Applied Biosystems 7500 fast. Now, this machine will at time will detect only 96 samples. One. Second, you need to first carry out RNA extraction manually, and then you can carry out the PCR part. Now, this can be resolved by much better machines like the one shown here. It is the Roche Cobas machine. In this machine, the RNA extraction as well as PCR as well as detection, everything starting from the swab is carried out automatically. So you need not to do RNA extraction. You need to just transfer VTM, virus transport medium containing swab, is to be given to the machine, and then the rest thing is done by the machine. So it is easy to use. One thing. Second, the number of tests which can be performed is in 1,000. So within an 8-hour of shift, technically presently all the countries, they are running in all the 3 shifts. The machines are working 24/7. But in an 8-hour shift, the number of samples which can be processed is more than 1,000. So this is known as the high throughput system. High throughput means the number of samples are high. Now, this is the common thing which is done today. But there are certain advancement in the RT-PCR technology. We call it self-enclosed rapid molecular assay system. Why self-enclosed? Because the tube is not open for the safety purpose. Entire thing is enclosed in a cassette, and the process is rapid than the earlier RT-PCR method. Now here what is done? So the regular qPCR machines are changed, modified with the research and innovation. So the original design is changed, and the PCR reaction is not carried out in PCR tubes or 96 well plates. It is carried out in what we call ready-to-use cartridges. And therefore, this group of machines are known as cartridge-based nucleic acid amplification test, or CB-NAT. Now, these cartridges are already having the reagents required for the PCR. That is all the DNTPs, buffers, primers, reporters, enzymes, everything is there in the cartridge. So you don't have to add anything. You need to just pour the sample, which is usually the virus transport medium. जिसमें आपने swab डुबाया, that virus transport medium has to be just pipetted into the cartridges. You need to place the cartridge in the slot in the machine, and the rest will be done by the machine. So, and the machine itself is also nowadays, with the research and innovation in design, The machine is made compact and portable so as it is available in even the small laboratories, even in the small towns. The classic case of such machine is by a company called Cifid Gene Expert, and Cifid, and the machine is known as Gene Expert. Now this machine is available even in Surat, so it is readily available in the small towns also in India. And the test performed for the COVID-19 diagnosis by this machine is validated and identified and recognized by the ICMR also. Now, what this machine is for? So originally, this machine was designed for the diagnosis of TB, and we have in India a TB eradication program known as RNTCP. Okay, and that is Revised National Tuberculosis Control Program. Originally, it was known as NTAP. Now, this program has set up laboratories all across India. So there are more than 1,000 laboratories in India which are having this machine, and the people, the laboratory staff is also knowing how to operate this machine. Now this machine, the company has just changed the cartridge, the reagents have been changed, and instead of regular TB cartridge, now you are having a SARS coronavirus 2 cartridge. So this is the virus causing the COVID. Just by changing the reagents, you can do this thing. Within the cartridge, if you see the anatomy of cartridge, then there are plenty of chambers. Each chamber is filled with the different reagents. So there will be RNA extraction in one, there will be reverse transcription in second, there will be PCR in third, and then the PCR reaction will be, the product will be checked in this protruded transparent part. In below, there is a valve, rotatable valve, which will push the liquid from one chamber to another chamber. So this is how cartridge and machine works. Now that is also expensive because it's from a originally the Cifid is not the Indian company. Now there is an answer, indigenous answer to that. An Indian company called Mol Bio Diagnostics from Goa. They have developed a system known as TrueNet system. It is very much similar, but the design is different. Now instead of one machine which does the RNA extraction and the RT-PCR, QRT-PCR, there are two machines. One doing the RNA extraction, the second one is doing QRT-PCR, and this is cheaper than anything else available, and it is already again identified by the ICMR. Now here the PCR reaction is carried out in a chip. There is a very small chip having microfluidized design, so the reagent will be flowing into the very small channels in the chip, and then you can see this white color part. This is the reaction chamber where the PCR will be carried out. All the reagents, primers which are needed for PCR reagent already are present there. Again, you need to just add the extracted RNA. Remember, the RNA extraction is carried out by a different machine. So you got a different machine which will do RNA extraction, and the extracted RNA is to be added onto this chip, and that will be read on a second machine. Now, this is a cheaper version of all the machines, and even the laboratories in India, there is a laboratory based in Indore in Madhya Pradesh. There are multiple machines placed to ramp up the speed of the testing, because as you know, we are doing thousands of testing. As a country, we have crossed many millions of tests. So, and everyone is pressurizing India to have the more testing. So in order to ramp up the testing, there are parallel machines being set up, and this is cheaper, low cost alternative to the other machines. Now, this is what we are doing presently in order to detect the RNA. But there are already many other future testing devices which have passed their design phase. I'll list down few of them. The one machine is known as Credo Vita PCR. Now, all these machines, they are doing still the same process, that is QRT PCR. Okay, but the advantage is that through the design, they have made cartridges which are simple. The tests are simple. The laboratory technician will only perform the process in less than 2 minutes. It's not a longer time. And then the final process, including the final result, is in less than 20 minutes. This is against the 2 hours in the regular process. So the time from 2 hours is reduced to the 20 minutes. The machine is smaller, so it can be used as a point of care testing machine, POCT machine, and it is cheaper also. The second machine is from a company called Bosch. This is Viva Lytic system. Now here again, there are cassettes. You can see 4 cassettes on bottom. Now there are many cassettes available for other respiratory viruses also. So there is an influenza A virus cassette, there is influenza B virus cassette, and the different cassettes will have different reagents. So what is to be done? Just scan the barcode on the cassette. Then you need to just pipette the virus transport medium into the cassette. There is an opening, and the rest is done by the machine. So again, less than 2 minutes of job by the technician, and the same machine can be used for many respiratory diseases. And the pinnacle of design is this machine from a company called Meso Biotech. It's known as Aquila system. Is one of the smallest PCR device. And here again, there is a cassette. What we need to do, we'll transfer our swab into a transport medium. So there is preparation stage of sample. And once you transfer the virus from the swab into the medium, from that, there's an ready to attach pipette. You just need to press the lead pipette and the reagents will be transferred to the cartridge. Place this cartridge into the machine and then the rest is done by the machine. Now, there are other machines which don't use the principle of RT-PCR. So we can detect RNA without doing RT-PCR. This is entirely different principle. And one such machine is ePlex system. Here, there is no PCR carried out. Rather, there is what we call the DNA-DNA hybridization. So the kit or the cartridge will already have a single-stranded DNA, which is complementary to the RNA of virus, and once the sample contains the virus, then the viral RNA will bind with the DNA, and that binding will be detected by the electrochemical method, what they call e-sensor. So the method is again very simple. It takes less than 2 minutes to the technician, and at the 8 hours of shift, you can carry out 96 tests. And there's a e-sensor. You can see the yellow color sensor pad where there is already the complementary strand, complementary then the virus RNA is attached, and then the binding of these two is checked by the electrochemical methods. There is another machine in evaluation stage, that is the microsense DX machine. Now here the DNA or the RNA of virus is converted into the DNA and then it is amplified. But the DNA amplification is not carried out by the method of PCR. The disadvantage of PCR is that in order to carry out amplification, you are having multiple cycles, and each cycle will have 3 different temperatures. So you need to first separate the double-stranded DNA by melting. The temperature is around 96. Then you need to attach the primers. So for that, the temperature is to be reduced somewhere around, say, 46, 45. And then there is extension by the enzyme Taq polymerase that is carried out at 72. Then again 96. So this cycle of 96, 45, 72, 96 is to be carried out repeatedly. Now that makes the machine complex. In this technology, the temperature is not increased or decreased. It is kept at common. So that is known as isothermal process. The temperature is not to be changed. Now this principle is known as loop mediated isothermal amplification or LAMP. In this, instead of one set of primer, there are 3 sets of primers. So within the DNA or RNA to be tested, there are 3 sets of primers at both the end, and then you are adding the repeated primers which are already there in system, and the amplification will be carried out with a terminal loop being created. And the DNA amplification there is no need of separating the 2 strands of DNA. It always remains single stranded. And then there is a reporter system which will detect the increase in concentration of the DNA. Now, this is known as loop mediated isothermal amplification. And there are many machines using this technique. Now, this is the way forward. No longer RT-PCR is to be carried out. There are advantages like the simplification of machine, plus it is rapid. So Abbott has came up with a machine with the same principle. Here you are having some consumables. The central blue piece is already having the media buffer. You need to place your swab here, then join it with the transfer cartridge, join it to the test base, and these 3 will be placed into the machine, and the machine will be doing the rest. So this is the process of Abbott ID system, and the same machine can be used for the diagnosis of other diseases also, because the buffer can be different, the reagents in the test will be different. Now the technology which everyone is looking forward to is a technology called CRISPR. Again, this is a non-RT-PCR technique. We are not doing RT-PCR. And what we can do, so we can use the enzyme system which can edit the DNA. So the target reporter DNA, there is a signal dye containing reporter DNA is broken up, but the breaking of that reporter will depend upon the presence of certain sequence from the virus. So if virus is present, the enzyme will be activated and the reporter will be broken. If virus is absent, then the enzyme will not be activated and reporter will not be broken. Now the enzyme is a system known as CRISPR-Cas9. So we are aware of this system. Earlier it was in research, but last week only a company has been given the approval for the commercialization of this test. The company is known as Sedlock. So Sedlock has got authorization for CRISPR-Cas based SARS-CoV-2 test. Here, what you need to do, so you are taking the virus, sample is prepared, and then the RNA is extracted. RNA is then amplified because you need to have larger concentration of sample, and then the CRISPR is activated and the reporter is broken in order to have the signal. Now, from the person's side, from the technician side, it is very easy in compared to PCR. It is faster than the PCR, and you can perform large number of samples. As you can see, it is a strip-based test. It is just using the strip and some reagents, so it is easier and it is really faster in compared to PCR. This we are looking forward. A very interesting new strategy is coming from the Israel, where a company called Nanoscent has developed a research platform where DNA is not checked, or sorry, the RNA of virus is not checked. Rather, the virus infection changes the pathophysiology or the physiology of human body, and certain volatile organic carbons are produced by the person who is been infected with the coronavirus, and we are being asked, the patient has been asked to just exhale into an enclosed bag. So आपको सांस बाहर निकालनी, you need to exhale your breath into the bag. The bag will contain then the gases from our body, and that gas will be detected in a detector machine for volatile organic carbon, and the test will take less than a minute. It is still in research phase in Israel and looking for the approval. If been passed, this will be a very rapid screening test. This can't be as accurate as RNA detection, but this can be for the screening. It is not for the definite diagnostic, but for the rapid screening of the patients. With this, we come to the rapid antigen test. Now, all this test will detect for the protein antigens of virus, specifically, as I said, a nucleocapsid protein called N protein. So, N protein is the target of detection. Already, these tests are being carried out in India, in Gujarat, in Surat also. And the first RCMR approved rapid antigen test is from a company called SD Biosensor. It's a collaboration between Indian company and the Korean company. In US also, there is a rapid antigen test now been authorized by US FDA by a company called Abbott. We have already seen Abbott RNA detection system known as Abbott ID, and now we are having the Abbott protein antigen detection kit also. It's a card-based kit. There is innovation in that also. For the safety of the technician and for the rapid detection and accurate detection of the card results, now there are machines which will identify the result in the card. So there is a slot where you need to add blood only, just 1 or 2 drops of blood, and the rest of will be done by the machine. It is very small machine, but very rapid antigen detection and protein detection is done. This is developed by a company called Celltrion in Korea. It has got US FDA approval for this test. Another company which has got its approval just few days back, 3 or 4 days back, is Lumira DX. Again, it's a rapid protein antigen detection machine, but the same machine can be used for many diseases. So just the strip will be changed, and you need to just place a drop of blood into the strip, place it into the machine, and the rest will be done by the machine. This will be detecting the virus antigen proteins. The last group of test is rapid antibody test. This will not detect the virus. This will detect the antibodies made by the human body against the virus. So this is not for the definite diagnosis. This will give you an idea whether you have been infected by the virus in past or not. So this will give you the infection history. All the antibody tests are carried out using such cassettes. Now the within cassettes there is a long strip, okay, lateral flow test, and that strip will have certain antigen and antibodies absorbed on the surface. As you pour the blood in the sample window, it has to be diluted with a buffer provided. You add 2 drops of blood, then 5 drops of buffer solution. This mixture will flow through this strip, and if the antibodies are present, they will be captured with the antigen already present in this test strip, then will be identified by an anti-human antibodies already been absorbed in the test strip, and you need to only observe the lines. So this principle is known as immunochromatography. It is just giving you the result in 15 minutes. However, the results are not of very high accuracy. There are chances of false positive and false negative in this method. Again, there are when the pandemic started, plenty of these kits of antibody detection were imported by the India from the Chinese company, but which are found to be not of satisfactory quality. And we have got now indigenous antibody detection test been made. One example is of Oscar Laboratories. So we are now producing this thing by our own. I'll come to the summary of entire thing. We'll first see the past of virus RNA detection. On 11th January, scientists in China released the complete genome of the virus. On the same day, in Malaysian Research Institute, the RT-PCR primers as well as probes sequence of that were released. So in 11th January, we are aware what we need, reagents, what we need in order to perform the RT-PCR. On the 4th of February, US FDA gave the emergency use authorization to the kits in USA. Earlier, the first well acclaimed, well accepted kit was from a company called Roche. Then the same technique of RT-PCR was changed, as I shown, with the different companies like Abbott, in order to have small machines, point of care machines, and that was approved on April 21. Presently what we are doing, so the home test of the RT-PCR using saliva or the very short nasal swabs is popular in the US, not really popular in India. Millions of tests are performed in US as well as in India, and scientists are looking for still modifying or refining this test in order to have very small machines, so as the entire RT-PCR with accuracy can be carried out even in small towns, even in the small laboratories. Future, we are looking for three things: validation of the already available home testing kits, development of CRISPR-Cas-based accurate diagnosis. Here, I would like to stress out that the company that I have shown, which has got authorization, is UK based, but in India, IIT Delhi scientists have developed the same thing that will be hopefully getting commercialized very soon. So we'll have CRISPR-Cas9 based test in India also. And lastly, there are other analytic machines under development to rapidly diagnose the virus RNA. Now, this is all about testing of the virus RNA. When we come to the serological testing of antibody, the first widely accepted test kit was from the Roche, which got approval in the March 3. And then the rapid antibody test, which got authorization in USA, was on 4th April. The problem in past was that there were plenty of test kits provided specifically from the China with non-satisfactory results, and therefore even in India, ICMR in March approved the use of antibody testing kit, and within the 1 month, ICMR retracted the use of antibody kits. Now, presently, again we are having good quality antibody testing, and therefore ICMR has again started using this same thing. Presently, scientists in research laboratories are trying to understand the behavior of memory cells and antibody role of antibody in the diagnosis and the therapy. In future, we'll be trying to have a more better understanding, a comprehensive understanding on the role of immune cells as well as antibodies. We'll also like to improve the accuracy and precision of the test. The challenge, the biggest challenge in front of us is that in India already we are opening up. We have already in the unlock phase. So now, as people will move around, there'll be more chances of more people getting infected, and therefore we need to have more testing. Now, the testing has to be affordable, rapid, and accurate. So this is the biggest challenge in front of us. Large number of testing, millions of tests, and these millions of tests are to be rapid, accurate, and affordable to all. That is our future challenge. With this, I will complete my presentation. Thank you all.
Thank you, sir, for wonderful presentation on the PCR techniques. It was a really knowledgeable presentation. Now we are going to question answer session. Audience, if they are having any questions, they can drop to live chat. So we will be raised to our speaker, Dr. Jay Bargi.
Now there is one question coming in, that what is CRISP R? So let me explain you the very short what is CRISP R. CRISP R is a biological process happening in nature which was originally detected in bacteria. This is bacteria's way of identifying the invading viruses. जिस तरह से कोविड वायरस हम लोग को इन्फेक्शन करता है, similarly there are bacterial viruses which will target the bacteria. So bacteria has got its own defense mechanism against this virus. So this is a system made up of enzymes and RNA. With the help of enzymes and RNA, bacteria will identify the invading virus, specifically the nucleic acid of that invading virus, and will break it. Now, what we have done from this natural process, we have taken up this system, enzyme and RNA, and instead of breaking the invading virus, now we can break any DNA. So, this is known as CRISPR-Cas9 system. Now, this is used in laboratory as a DNA editing system. This is used to break the DNA. So, what we do, we take a reporter probe, which is giving you the radiation light, not when it is intact. It will give you the light when it is broken. And the enzyme will break that reporter DNA only when it is activated. Now, the activation of enzyme demands the virus RNA. So, if a patient has got virus, there will be presence of virus RNA. That virus RNA will activate DNA breaking enzyme, that is CRISPR-Cas9 system. Once activated, it will break your reporter probe and you will be having signal which can be observed as a color change. So technically, this can be used as a diagnostic tool. Originally, it was defense mechanism of bacteria. That is what CRISPR-Cas9 is. Now, there's a question that if you want to understand that what is the currently authorized method, then which is the site available. So in India, I would recommend I do it almost on daily basis. You need to visit the ICMR site, and ICMR is Indian Council of Medical Research, and they've got their dedicated COVID-19 page. On the COVID-19 page of ICMR, you'll be having all the information regarding not even the present but even the past validated methods and machines. So what ICMR does, suppose there are 13 companies which are willing to sell the diagnostic test kit in India. So what ICMR will do, it at its own will carry out some sample testing on those 13 kits, and then it will approve the kit with the very high standards of accuracy and precision. And I have seen reports where as many as 13 kits were evaluated, including the kits from the USA, Singapore, South Korea, and only one was given the authorization. So ICMR has got very stringent screening procedure of identifying which is the right kit. In India, you should ideally read the ICMR COVID-19 page in order to get information regarding which kits are authorized, which methods are authorized. Even they have given the SOPs, the procedure of using the kit. So all these informations are publicly available on ICMR page. I would sincerely request all of you having any question in the ICMR for this. Now, sir, Vikram sir has already given you the feedback link for the day one. What you need to do, you need to fill in the feedback for both the speakers, and then tomorrow, same time, 10 o'clock, we'll have two more sessions. One is regarding the pathophysiology of the virus, how virus causes disease in human body, and the second one is regarding how the spread of virus is concerned with the environmental response. So we'll have these two sessions. Again, there will be feedback, and then we'll be surely giving you the certificates. We are not holding back the certificates. Certificates will be issued surely for all the participants. But once you give the feedback for day one as well as day two, day one feedback link is there. Any other question? Now, when you go for the antibody testing, is it possible that polyclonal antibodies in serum of patient will give you the false? Yes, there are high chances of false positive test, and therefore antibody detection is not considered as a diagnostic test. In India or in any other global authority, detection of serum antibodies is not considered as a diagnostic test. That is the reason because it gives a large number of false positive as well as false negative results. So antibody detection will just give you the hint about your own immunity against the virus. Is to see that in a given community, say for example in Surat, what is the number of people already immune against the virus, or what is the number of people, proportion of people, percentage of people which are still at the risk of virus. So if your antibody test is negative, you are still at the risk of virus. If your antibody test is positive, you are now immune to virus. और इसको हम large sampling करेंगे, then it will give us the idea about the community. So Surat is at what percentage of number of population is having immunity against the virus or is at the risk of virus. For that only antibody testing is done. It is not for diagnosis on account of false positive as well as false negative results. Yeah, one question is regarding the specificity of CRISPR-Cas9 test. Now, CRISPR-Cas9 test is seriously theoretically sequence dependent. So the specificity of CRISPR-Cas9 test is very high. It what it does that it goes for the region of the virus protein which is not mutating very rapidly. So when we say virus is mutating, not all proteins of virus are mutating at the same speed. There are proteins of virus which are conserved. There are proteins of, and within the protein there are again small parts, what we call domain. So there are domains in virus which are conserved. What we do, the RNA sequence against this conserved domain, we use that. For that reason the diagnostic test will last for longer time. It's not going to be क्या आज एक नया virus, today there is a mutated virus and it will not be detected by our diagnostic test. It's not going to happen because the domain selection of the virus. So usually there are two domains. One is the ORF1AB, the second one is N domain. So these two genes are targeted for the CRISPR-Cas9. The accuracy of CRISPR-Cas9 is beyond 95%, and it is giving very satisfactory result. It has been tested in US, Europe, and in India. So the results of CRISPR-Cas9 are going to be definitive diagnostics. Now, one question is regarding how many samples you can test. The sample testing is not the limiting factor. The limiting factor is the availability of CRISPR-Cas9 consumable. The more strips you are having, the more samples can be tested. It's a strip-based assay. Just you need to place the virus medium, that is the transport medium, and the results is seen. So the sample processing is not going to be the limiting thing. One question is very simple: RNA or DNA, which is detected in virus? So virus is saying only RNA. There is no DNA with the virus. But for detection purpose, in our test tube or in our cartridge, we convert RNA into the DNA. So when we detect the DNA, it is made from the virus RNA. So actually, we are always detecting RNA. We can't detect DNA because there is no DNA with virus. Is the rapid antigen test more better than the PCR or not? So presently speaking, the gold standard for the diagnosis of coronavirus, SARS coronavirus 2, is the RT-PCR. The antigen detection is, yes, definitely it is giving you the result. The result is validated, is been considered authentic by the governments in US as well as in India, but it is not considered better than the detection of RNA. It is never considered better than the RNA detection. It is at par with the RNA detection. Now let me tell you, there was a news item. The question is asked by someone that a new super spreader strain has been detected in Malaysia. Now I myself has tried to dig into the thing, and what I have found that all the news platforms in India, like there was a platform, I would not like to name any of the news platform, but all the news platforms in India had this news of a new deadly strain of virus detected in Malaysia. Their big resources was one international article, and that one international article from which this news was taken up never explained the complete biology of this thing. And when I went into the scientific publication of the scientists which were named, then this is not a super spreader virus which is not be controlled by the vaccine. There is no such things. So technically, this entire thing regarding Malaysia having a new virus which is super spreader can't be controlled by vaccine. This is a hoax. This is not the true news. So the Malaysian strain of virus is still under the ambit of vaccination, and it is not 10 times deadlier as been reported by many news platforms. I would sincerely advise to all the people that you are students of biology, you are students of science, kindly don't go with the normal news platforms. You read the correct information from research articles or from the ICMR website. These are the hoax news that there's a deadly virus in Malaysia, 10 times deadly virus. There's no such thing as that. So I think that suffices the question answer, and there's no new question coming up. So I would again at the end of day one, I would thank all the participants who are present here. Uh, we'll be posting the link again and again. If not working, then we'll be even emailing the links. There's no issue about that. The link will work. You will be able to find the feedback. We'll resolve those things. I'll thank all participants which are present here. I thank the first speaker, Avni Madam, and I also thank Vikram sir for moderating the technical side of this webinar. And we will meet again tomorrow at 10 o'clock sharp with two more speakers. And at the end of that, we'll be giving you the certificates. That's all for today. I thank all of you.
Shall we over it, sir?